In this video, we will discuss about whether the GlyTR1 can overcome the immunosuppressive mechanism of tumor microenvironment or not, and we will see the results uh, from the in vitro studies and also from the in vivo studies. So we will start with the in vitro studies. So first, they check the GlyTR1 roles in the presence of GlyTR1 roles in the presence of L-PHA lectin. So what it is, this is the GlyTR1 has a domain that can act as L-PHA. So they used L-PHA alone, and on the other hand they use GlyTR1 uh, along with this ovarian cancer cells and check the cell death. What they are able to find out, in the L-PHA alone they are able to see the cell death, but at 100 folds higher concentrations compared to the GlyTR1. And one of the probable reasons could be because the GlyTR1 also has anti-CD3 domains that can also helps in the cell death. And for this tumor microenvironment, what it is, they suppress beta-1/6 Glycan branching that is detected by GlyTR1, and they are able to see reduced in the cell death accordingly. That can be seen here, where the when the beta-1/6 Glycan is present in high amount on the cell surface, the cell death is the cell death is high, whereas when the beta-1/6 Glycan is reduced, the cell death is lower. That can be seen here in this black line results in the panel B. And in the when the beta-1/6 when the MGET1 is reduced, it will also reduce the cell death, as it will also affect beta-1/6 uh, branching in the tumor cell surface. And when they check it about this in the triple negative breast cancer cells, when they check in the triple negative breast cancer cells, the effect of this GlyTR1 and T cells, and only the T cells, they are able to see that when there is only T cells are there, there is no cell death that they have observed. But when GlyTR1 and T cells are both present, they are able to see maximum cell death. Even in the presence of cytokines, that is interleukin 10 and TGF-beta, which is also a type of immunosuppressive factors. Next, they check the effect of these T regulatory cells, which can also act as immunosuppression. And they can see that with up to about 20% T regulatory cells, they do not see any significant effect on the cell death. But when they used around 75% T regulatory cells, they are able to see some effect. However, this is still higher compared to the case where only cancer cells are present. And when they check the about effect of hypoxia, they are able to see that the effect that when this ovarian CSCs and the breast CSCs, cancer stem cells, they are able to see the cell death in the hypoxia conditions, even in the presence of GlyTR1 at different concentrations. That can be seen here. They can even in the concentration of like picomolars, they are able to see a cell death even in the hypoxia conditions. This indicates that GlyTR1 can overcome most of the immunosuppressive mechanisms or immunosuppressive factors in the tumor microenvironment. Once they validate their results or they have the positive outcomes from the in vitro studies, they wanted to check it in the from the ex vivo studies. They wanted to check it the results in the in vivo studies. And so far from panel A to E, the results experiments are done in the in vitro studies in the in vitro environment. So for the panel F, what they did is they took the clone 11, which is the MGET5, and co-culture them in the breast cancer affected mice. And so what they did is they took this triple negative breast cancer tumors in which MGET5 has been knocked down, and cultured them in the mice, and then they took out some of these cells and make an organoid out of it, and also add GlyTR1. And what they are able to see, that in the case of this MGET5 knockdown, the cell death is around 13%, whereas when the MGET5 is not knocked down, the cell death is around 55%, indicating that the cell death is dependent on the beta 16 Glycan branching, and the live cells and dead cells are also shown here in the quantitative forms, or the to show the its reproducibility, that in the case of beta 2 they are able to see more live triple negative breast cancer cells compared to the case where MGET5 is not knocked down. Thereafter they check this breast cancer cells along with this PBMCs, and what they are able to see, that when they use the single cells suspensions, when they use the single cell suspensions, they are able to see around 22% of the cells are these PBMC cells, and rest of them are tumor cells. And among that, they are able to find out that some of the T cells are also present, indicating its infiltrations. And then they check it about within these T cells the presence of the PD-1 ligand. So around 89% of these T cells has. So first, from the tumors, they wanted to see whether there is infiltrations of the immune cells or not. So in the tumors, they are able to find out 22% of these PBMCs. In that, they are able to see 99% of the T cells, and in these T cells, they are able to see 89% of these T cells has this PD-1 ligand that can act as an immunosuppressant, or they can suppress the acute T cells. And within this 89%, they are able to see different populations of the T cells, that is CD8, 2.2, CD8, and CD4 plus percent. And this indicates that some of the cells are still in the most of these cells are in the form of like deactivated form. However, when they take the tumors and form an organoid and use the GlyTR1, after 7 days they are able to see the triple negative breast cancer cells are around 49%, and in the case of control, whereas in the case of GlyTR1 they are able to see triple negative breast cancer cells are around just 14%, indicating that triple negative breast cancer cells, or this GlyTR1 functions well on this triple negative breast cancer cells that has the beta 16 Glycan branching. And the first row is shown about the tumors expressions, how much is the immune cells are present in the tumors, but they are not performing or they are immunosuppressed, we can say like that. Next, they check the patient samples. That is, take the tumor samples from the patients and use the singles. So for the panel G is experiment done on the mice. Next, they did their similar studies on the patient's tumors. So when they check the single cell suspensions, they are able to see around 7.1% are white blood cells in the tumors. Among that, 13.4% are the T cells, and in that they are able to see the exhausted cells are around 58%, and within that they are able to find out that 70.4% are in the lung. And then when they check its effect with the GlyTR1, they are able to see that. So we will discuss this in the while. So the tumors image are shown here, that HNE and vimentin. Vimentin is, you can say, a proliferation markers that can show the tumors are spread about. Then what they did is they take the tumors and use the GlyTR1, the similar experiment that they did for the mouse, and then culture them for 6 days and digest in the then they did the 2D culture, and in the case of control they are able to see most of the tumors, but in the case of GlyTR1 they do not see any tumor cells, and the quantification shows that when the GlyTR1 is not present they are able to see the live cells, live tumor cells, but when the GlyTR1 are present they are not able to see, or presence of live tumor cells is significantly lower. And this figure indicates that when the tumors alone are present, or uh, the tumors alone are present, and the amount of GlyTR1 required is very high. And however, here this is the control, that is CSV plus. Here also we can see the GlyTR1 requirement is higher. However, when the case where uh, tumors are, in this case tumors are co-cultured with GlyTR1, the amount of GlyTR1 then so here what is shown here is that when the tumors are alone the GlyTR1 requirement is high. When the CSV plus they are able to see it is kind of a control the GlyTR1 requirement. And in the 2 only, 2 only is the I think is the conditions where they are co-cultured in the absence, where they are co-cultured in the absence of this beta 1,6 branching, beta 1,6 and Glycan branching. I'm not right now remember what this 2 degree only means, but if that is the case then we can see that the GlyTR1 amount required is lower, and this could be because the anti-CD3 can act on the tumor cells. So I hope you understood how this they have validated their GlyTR1 results and on this tumor microenvironment factors, different tumor microenvironment factors. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.